Meet the Scientist monthly webinar series. I'm Dr. Jeff Borenstein, President and CEO of the Foundation, and your host and moderator for today's webinar. Today, Dr. Wade Berrettini will present novel findings in brain DNA for psychiatric disorders. The Brain and Behavior Research Foundation funds research around the world that identifies the causes, improves treatments, and will ultimately result in preventative techniques and cures for mental illness. Since 1987, the Foundation has awarded more than $324 million in research grants. We are the largest private funder of mental health grants other than the federal government, and what makes us unique is that 100% of all donor contributions for research are invested in the grants given directly to our researchers. The researchers are working to find scientific breakthroughs in brain and behavior disorders such as ADHD, anxiety, autism, bipolar disorder, borderline personality disorder, depression, obsessive-compulsive disorder, post-traumatic stress, and schizophrenia. I'm delighted to introduce Dr. Wade Berrettini. Dr. Berrettini is the Carl E. Rickles Professor of Psychiatry and Director of the Center for Neurobiology and Behavior at the University of Pennsylvania Perelman School of Medicine. Dr. Berrettini's research interests include the genetics and pharmacogenetics of several complex conditions, including addictions, mood disorders, eating disorders, and epilepsy. Since 1997, he has served as the director of the Center for Neurobiology and Behavior, a group of interdisciplinary scientists studying genetic and biologic influences on brain disorders and behavior. Dr. Berrettini earned his MD degree and his PhD in pharmacology from Thomas Jefferson University. After completing residency training in psychiatry, he studied the genetics of behavioral disorders at the National Institute of Mental Health for 10 years. He was the recipient of our NARSAD Distinguished Investigator Grant and numerous other NARSAD grants, and the Bipolar Mood Disorders Prize for Outstanding Achievement in Mood Disorders in 1996, renamed the Colvin Prize in 2012. He is also a member of the Foundation Scientific Council that identifies the most promising research ideas to fund with Foundation grants each year. Today's webinar will start with Dr. Berrettini's presentation. This will be followed by question and answer period. To submit your questions, please use the questions tab on the control panel on your screen. You can submit your questions throughout the presentation, and following the presentation, I'll present your questions to Dr. Berrettini, and he will address as many of those as possible. And now, I'm pleased to present Dr. Wade Berrettini. Wade, the floor is yours.
Oh, thank you, Jeffrey. Can people see this first slide?
Yes. Yes.
Yes, perfect. Yes, thank you.
Terrific. Okay. So I'm going to discuss a little bit about very recent results out of our lab where we're actually looking at the DNA of nerve cells from post-mortem brain to find DNA sequence variations, sometimes called alleles, which might predispose people to psychiatric disorders. And I'm going to share with you results from a study in schizophrenia and a study in cocaine addiction. And if there's some technical term that puzzles you, please ask me through the webinar mechanism to explain it. So this kind of research is quite different from genetic research, which NARSAD and the Brain and Behavior Research Foundation have supported for quite a few decades. typically in that kind of research, scientists take DNA from white blood cells and they study that DNA under the assumption that brain DNA has the same DNA sequence. We now know that in many instances this isn't true, and this falls under the category of somatic DNA variation. And this refers to DNA variation that occurs during development, so that these types of variants are not present in all the cells of an organism. The earlier in development a variation occurs, the more widespread its distribution will be in the organism. And I have an example of this at the bottom of this slide. So you can see this developing neuron here in the embryonic brain in utero, and as this cell gets ready to divide, it has to copy its DNA so that each of the two daughter cells will have a complete set of chromosomes. And as you can imagine, sometimes this is not done perfectly, so variation can creep in, as indicated by this little red circle. So at the end of the production of two daughter cells, you can see that the two daughter cells differ by a small amount in their DNA structure because of the error that occurs in replication. Now, we're going to talk a little bit about the opportunity for these kinds of DNA variations to creep into brain nerve cells. And this relies on the fact that in the developing embryo, there are just a few hundred cells that give rise eventually through development to the 170 billion cells of the adult human brain. And these 175 billion cells are equally divided between neurons and another type of cell that we call glia, whose role is to support the neurons in their activity. Somatic DNA variation has been studied for decades by cancer researchers who would take tumor tissue and examine the DNA and find all kinds of variations in the tumor tissue that were not present in the adjacent normal tissue. And this has given rise to the concept that as a tumor develops, these somatic DNA variations may cause the tumor to become more aggressive and spread more rapidly, or metastasize as we call it, or develop resistance to chemotherapy medications. An important point here when we apply this concept of somatic DNA variation to psychiatric illness is that these types of variants will not explain why these illnesses tend to run in families. That is, they will not explain what we call heritability. This type of somatic DNA variation will only be detected, for example, if scientists can study the DNA from neurons and glia. And that's why it's so important for the country as a whole to have organized what we call brain banks, where people with and without these illnesses can sign up to donate their brains upon death. That's such a key part of our ability to make progress in this area. So now I want to introduce a concept of a kind of viral DNA sequence called long interspersed nuclear element 1 retrotransposons that these represent one mechanism by which somatic DNA variation can occur both in the brain and in peripheral tissues. Here I have to get a little technical, and I apologize for this. But it's long been known that these line 1 or L1 DNA sequences, which are shown in this slide in the middle, and there are about 6,000 base pairs of DNA in length, that they exist in the human genome. And these are related to ancient viruses that have incorporated their DNA into our genomes, and they spread in our genomes via copy and paste mechanism. And these kinds of retrotransposon sequences, when they go into this copy and paste mechanism, can insert into a gene which is needed by a cell for normal function. And we can see in the history of medicine over the past 20 years that there are about 100 examples of different diseases caused by these retrotransposons disrupting a gene that's needed by the cell. And there are also several examples of these retrotransposons causing human diseases through somatic DNA variation as opposed to disrupting a gene in egg or sperm cells. These retrotransposons undergo a complex life cycle where the L1 DNA gets made into messenger RNA. The messenger RNA gets transported out of the nucleus into the cytoplasm where its proteins are made. The messenger RNA and the proteins complex to re-enter the nucleus, and there the messenger RNA gets of the retrotransposon gets remade into DNA, and then the host DNA repair mechanism allows it to integrate into the human genome in a new spot. And that's how this copy and paste mechanism works. And if the new spot falls into a gene that's needed by a neuron for normal function, why that can represent trouble or risk for perhaps a psychiatric disorder. And it's this kind of idea that we wish to test both in schizophrenia and cocaine addiction. So you might ask, well, why should we study these two quite different diseases with this retrotransposon concept? Well, that's because for both these diseases, studying white blood cell DNA has not been wonderfully successful in allowing us to understand these disorders in a more complete way. And so we thought maybe we can make better progress by studying the DNA of the nerve cells themselves. Here are a few other kinds of diseases where studying L1 retrotransposons helped in understanding the complexity of the disease. And so since these diseases were better understood by studying these line 1 retrotransposons, we thought to do the same thing. There are a couple of studies already in the literature connecting these L1s to the action of cocaine. For example, one group showed that if you gave cocaine repeatedly to mice, you saw an increase in L1 messenger RNA in the brains of those mice. And the Japanese group, Okudera et al., showed that if you took cultured nerve cells and incubated them with cocaine or methamphetamine, you could see an increase in L1 retrotransposition in those cultured nerve cells. So we wondered whether something similar was happening in the brains of people who were addicted to cocaine. So this experiment relied on our wonderful collaborator Deborah Mash at the University of Miami, who's been collecting the post-mortem brains of people who died of cocaine overdose for more than a couple of decades in cooperation with the Dade County Medical Examiner in Florida. She sent us 30 pieces of medial prefrontal cortex from persons who had died of cocaine overdose. And these persons, according to information from next of kin, had long histories of cocaine addiction. So we homogenized these pieces of medial prefrontal cortex along with 30 prefrontal cortex samples of accident victim controls, and we made preparations of the nuclei of all of these different nerve cells. And we were able to isolate by antibody staining the nuclei from the neurons and separated them from the nuclei of the glia so that we could just separate and study the neuronal nuclei. And here's a picture of what these neuronal nuclei look like under the microscope when stained with this particular antibody. So using this fluorescent NuN antibody, we were able to separate the neuronal nuclei from the glial nuclei using a process known as fluorescence-assisted cell sorting, or FACS. Fortunately for us, this worked quite well. Here you can see the separation of the NuN positive from the NuN negative nuclei, the NuN positive being the neuronal nuclei, the NuN negative being the glial nuclei. So then we did this complex DNA amplification procedure on the neuronal nuclei using the DNA inside those nuclei, and we sent the amplified DNA for DNA sequencing using this technological advance over the past, say, 5 years of high throughput DNA sequencing. And this process works remarkably well, and the DNA sequencing people returned to us about 500 billion bases of DNA sequence for us to analyze from the cocaine-addicted post-mortem brains and the control post-mortem brains. And to do this analysis of the DNA sequence, we relied on a wonderful collaborator at Johns Hopkins, shown in this photograph, Haig Kazazian, and his graduate student, Tara Doucet. They had developed this L1-seq program that allowed us to do the sequence analysis. And so we searched these DNA sequences and found quite a few L1 retrotransposons not previously observed in human DNA to be disrupting genes that were brain expressed among the cocaine-addicted people, but not among the controls. And this was particularly evident when we did some pathway analysis And this slide here briefly explains what we mean by pathway analysis. So over the past 50 years, various biomedical scientists have discovered that a vast array of interactions among proteins in human cells. So there are literally thousands upon thousands of different proteins in each human cell, and many of them interact with each other. And these millions of interactions have been cataloged and recorded in computer programs so that these interactions define signaling pathways in cells. An example of which I've included here. So we know from biochemical studies that protein A binds proteins B and then C, and so that's a signaling pathway in a cell. So we used computer-assisted pathway analysis to help us understand whether these line 1 retrotransposons that had never been seen before in human DNA were randomly landing in our genome, in the genomes of the cocaine-addicted brain samples, or whether there was some pattern to their appearance. And the pathway analysis, which uses this program called David, actually revealed something quite striking, which was that the brand new L1 retrotransposons landing in genes needed by neurons tended to land in genes relating to protein phosphorylation, as shown in this slide. So you can see here the different categories of pathways that were disrupted by these line 1 retrotransposons, and they're all related to phosphorylation in the cell. This term kinase refers to an enzyme which transfers a phosphate group onto a protein, so that all of these pathways share one principle, that they're involved in protein phosphorylation. So what was remarkable was none of these pathways were identified in the control post-mortem brains, suggesting to us that repeated use of cocaine damages all of these signaling pathways in the brain that use phosphorylation as a signaling mechanism. And the importance of this is that we may now be able to better understand the progression of cocaine addiction and can use this information to develop new treatments. Unfortunately, there is no FDA-approved medicine for the treatment of cocaine addiction. So here's an example of a pathway that was disrupted at multiple points by a new, previously undescribed L1 retrotransposon, and it's the fibroblast growth factor 2 pathway, here abbreviated FGF2. So it has its receptor and this signaling pathway that has 2 arms to it, and you can see that many of the genes were in this pathway were disrupted by these previously unseen L1 retrotransposon events. So this pathway then might not be a bad pathway to look at for developing new treatments for cocaine addiction. And incredibly, many of the genes in this pathway have been identified as having a role in the action of cocaine in previous biomedical research. And I list a couple of examples here. And so we think then that we've identified a mechanism whereby repeated cocaine use disrupts genes in neurons that may have something to do with the action of cocaine in the brain. And this is another slide with some additional genes that have been disrupted by L1 retrotransposons and have been previously implicated in the action of cocaine. So, and here's a third slide listing a lot of kinase genes that similarly have been disrupted by L1 retrotransposons and previously identified as playing a role in the action of cocaine. So there is one caveat to this research. We think that cocaine is acting in the brain to allow these L1 retrotransposons to spread, but we can't completely rule out the idea that all these retrotransposons developed in these individuals and acted as risk factors for cocaine addiction prior to any exposure to cocaine. And so we hope to be able to conduct additional experiments to distinguish between these two possibilities. However, the existing evidence suggests that these L1s arose as a result of chronic cocaine use over years. And if that's true, it suggests to us that repeated cocaine use disrupts neuronal DNA in a way that may explain why so many people with cocaine addiction have terrible cognitive difficulties, as well as trouble with depression and other behavioral difficulties. So in summary then, what we have here is evidence that chronic cocaine use disrupts a lot of signaling pathways in the brain that perhaps are activated by cocaine, and this, if confirmed in additional research, would shift our thinking about cocaine as an addictive substance, because now we would know that using cocaine may, in an irreversible way, change the DNA of our neurons. Now, I'd like to shift from the cocaine study to a very similar study that used dorsolateral prefrontal cortex of people who died with schizophrenia and matched control tissue. So we did the exact same nuclear preparation and fact sorting and DNA amplification steps that we went through in the cocaine study, but this time we did them in the using these brain samples from people who died with schizophrenia. So so now we get to the pathway analysis of this new experiment between tissue samples of people who died with schizophrenia and matched controls. And here we can see that the pathways identified are completely different from the cocaine study. They have nothing to do with phosphorylation, but they have to do with synapses. And here you can see the p-values, and they have to do with how nerve cells project and organize and develop in relationship to other nerve cells. And again, none of these pathways were identified as significant in the controls, and suggest to us that the genes involved here are important in the development of schizophrenia. So while we were doing this research, another group from Japan did something quite similar, using 3 post-mortem brain samples from people who died with schizophrenia and 3 controls. And I'm showing their results and comparison with ours. And even though they used somewhat different technical approaches, our results were remarkably similar to theirs. Identifying the same pathways, synapse part and plasma membrane part, as disrupted in the samples from people with schizophrenia. So that leads us to think that that these results are really quite reproducible across laboratories, but we wish to study this phenomenon in a larger number of post-mortem brain samples of people with schizophrenia and matched controls to convince ourselves that this line of investigation should be pursued and would yield useful information regarding the origins of schizophrenia. Again, one of the things that surprised us was that many of the genes in these pathways had been studied before and found to be important in the development of schizophrenia, and I list some of them here. So that this is another piece of evidence that leads us to believe that this line of research may be helpful in furthering our understanding of schizophrenia. And this is another slide showing some of the genes that we've identified have been studied in the recent past by other laboratories interested in understanding this complex disease. So if we were to compare the results with schizophrenia and cocaine addiction, we can see that they're really quite different, and this leads us to conclude that studying these line 1 retrotransposons in DNA from neurons of brain can tell us something specific about different brain diseases. And so we hope to continue this line of research in studying post-mortem brain of people who've died with terrible recurring depression, as well as people who've died from bipolar illness, and to compare the results with schizophrenia and cocaine addiction. So by way of summary, we've established that somatic neuronal variation in DNA may be a common source of non-heritable risk for these brain diseases. Evidence from the schizophrenia and cocaine addiction projects indicate that similar studies should be started in diseases like autism and depression and bipolar illness, as well as other addictive disorders. We should also consider additional studies in other kinds of brain disorders like epilepsy. The most important point will be that all of this research will require large, well-characterized post-mortem brain collections, and that's why it's so important for everyone, independent of their disease status, to consider donating brain for research after death. So I'd like to thank you for your attention. These are the people who worked with me on this project, without whom none of this could have been done. And I'd also like to thank the National Institute of Mental Health, and the National Institute on Drug Abuse for their grant support that allowed us to do these studies in schizophrenia and cocaine addiction. So with that, I'd like to close, and I'd be very willing to answer questions from everybody using the remaining time. Hello.
Dr. Borenstein will be on the line in a second.
Hello. Yep.
Okay.
Hello. Hi.
Yes.
Hi. Wade, thank you very much for an excellent presentation about extremely important work that you've been doing. A number of the questions that we've received relates to the issue of non-heritable risk versus inherited. Could you give a little bit of information about those differences between, what gets inherited that we think of inheriting blue eyes versus other types of genetic risk?
Sure. Remember that eggs and sperm unite to produce a developing embryo, and any DNA variation in an egg or a sperm that unite to produce a developing embryo will be distributed in all the cells of that organism, and that's the including the eggs and sperm of that eggs or sperm of that developing person. And that's the kind of risk that's inherited. So if the DNA variation is in the egg or the sperm, it will get passed on potentially to subsequent generations. And that's heritable risk. The kind of risk we were discussing today is non-heritable in the sense that it develops not in the eggs or sperm, but in brain cells while the organism is developing. And this happens in all kinds of mammals. So this is DNA variation that may predispose a person to disease, but it's not passed on in families because that DNA variation is not in the eggs or sperm. I don't know if that helps.
Yes. No, that's very helpful because I think that, as a general rule, people, lay people in particular, hear genes and think of inheriting blue eyes or blonde hair. And there's been a lot in the press about the potential of personalized medicine and using genetic testing to help pinpoint treatment for individuals. How does that fit into the types of things that you've been researching and just sort of the broader picture of genetics?
Well, certainly the promise of personalized medicine is so very important for the brain diseases that are the focus of the Brain and Behavior Research Foundation. And while we might say that this group of disorders is called bipolar illness, that group of disorders is very heterogeneous, meaning what looks the same to us clinically may have very different origins in terms of the person's biochemical makeup. And so the task ahead of us is to find the subgroups within, say, all people with bipolar illness or all people with schizophrenia who are going to do well on a particular medicine so that they get the best treatment at the earliest possible time. And that's the promise of personalized medicine. And this kind of DNA research can be an important step in getting there, but it's only the first step.
I think that's a very good explanation, and basically what you're saying is what we now call bipolar disorder in actuality is probably bipolar A, bipolar B, bipolar C, and as we are able to learn more about the genetics, we can better differentiate it, and A responds to this and B would respond to that.
Exactly.
With regards to the specifics that you presented in terms of your work with cocaine, how would you be able to differentiate whether or not this issue with the phosphorylation is a result of the cocaine use or, as you described, the possibility that there was a risk factor for cocaine addiction and the person may have had a predisposition towards this?
Yes. Well, one of the ways we could do this is by studying mice. Mice have their own L1 retrotransposons, and we could administer cocaine repeatedly to mice and then look in the brains of the mice for this kind of increase in L1 retrotransposons in these kinase phosphorylation pathways, and compare those results with mice treated with like salt water.
Okay, so you would be looking for the differentiation of being of the mice being exposed to cocaine versus, you know, being exposed to placebo.
Exactly.
And in the work that you're doing with schizophrenia and the effects upon synapses, this would be something that is more inherent in the, it's not due to some environmental effect, it's something that is occurring in those individuals without that kind of, you know, cocaine you could look at as an environmental effect, something that's being done to the brain. The synapse effect that you're finding in schizophrenia, that's not associated with an environmental issue?
Well, we know very little about it, Jeffrey. Our thinking is that this kind of line 1 or L1 retrotransposition can occur whenever there's abnormal electrical activity in the brain in a repeated fashion. So to take the example of cocaine addiction, every time a person takes cocaine, there's a wave of highly abnormal electrical activity in the brain, and we think that that opens the window for these L1s to spread. In the case of schizophrenia, we have, for about the past decade, been aware that in the brains of people with schizophrenia, there's this abnormal electrical activity occurring almost all the time called gamma oscillations. And it represents a certain pattern of electrical activity that can be observed on the EEG, or electroencephalogram. Our current hypothesis, and it needs a lot of support, is that this abnormal electrical activity in the gamma oscillations, which may be a risk factor for schizophrenia, may be opening a window of opportunity for these L1 retrotransposons to spread in the brains of people with this disease.
So that in theory, at some point down the road, in people who may be at higher risk, if there were an intervention, whether it be medication or some other intervention, that would decrease the likelihood of the L1 retroposon spreading, then that would be an intervention that might be able to stop the progression or even avoid the initial occurrence of schizophrenia in an individual.
Exactly. That's a good way to put it, Jeffrey.
Okay. And One of the questions that somebody asked, and relates to something we discussed earlier about, the different versions of bipolar, is that people with different psychiatric diagnoses may have similar types of symptoms, and the question relates to how does the symptoms that someone has, separate from the diagnosis that we use, how does that potentially relate to this type of approach of research?
Well, unfortunately, the individual symptoms, say of schizophrenia or bipolar illness, have not to date allowed us to create, let's say, uniform and informative subgroups of these illnesses that have clearly been responsive to a given treatment. So that the symptoms themselves haven't been very useful in creating subgroups of these disorders. Does that answer the question?
Yes, I think so. Another question that a few people asked relates to where you see this going. What do you expect will result from your research and others doing similar type of work in the next 5 years, the next 10 years? What can people expect with that?
Well, we are hopeful that this pathway analysis has highlighted groups of genes that should be studied in both cocaine addiction and schizophrenia, with an eye to developing new medications that may address the genes disrupted. And in this way, we are hopeful that the work we're doing today will be just the start of new medication development, because certainly some of the genes we've identified in both cocaine addiction and in schizophrenia are what we call druggable targets. That is, the pharmaceutical industry knows these genes well, knows how to design small molecules, potential medicines, that will interact with the proteins produced by these genes in predictable ways. And that's key to using this genetic information to make new medicines.
That's certainly very, very hopeful for people. One of the issues in an illness such as schizophrenia and bipolar, as well as often for cocaine addiction, is that the illness begins to take hold in late adolescence, early adulthood. And how does the work that you've been doing relate to this issue of the age of onset of these conditions?
There's no information that we have in the current schizophrenia or cocaine addiction project that helps us understand age at onset, unfortunately. Our results are really quite new, and the first step is, to do independent confirmations in new groups of samples. We haven't looked, we don't have enough results right now to try to look at whether the pattern of L1 gene disruption has anything to do with the age at onset.
Okay, so really this is what you're describing to us, which is part of why it's so exciting for everybody to be a part of hearing what you're doing, is very cutting edge, and is just the beginning of this area of pursuit. So that level of detail is something that's one of the questions down the road. Hopefully, you'll be able to begin to answer.
Yes, we certainly hope so. I think it will take many more than, say, 20 or 30 samples to help us understand whether this at all relates to age at onset.
Right. Just as an aside, one of the people who asked a question or a comment said that he or she has bipolar disorder and would like to be able to, when they pass away, have their brain available for science, which obviously is very important for these studies. So it's nice to see that people want to do anything they can to be helpful in this process.
I can't emphasize enough the importance of people giving consideration to brain donation to help us in this work, because without their participation, we will get nowhere.
I think that's just so important. It really is a collaboration between the scientists and the people and their families who are affected by these conditions. For the group that you studied with schizophrenia, is there a way that you could differentiate between the effects of the illness itself versus the effects of some of the treatment that the people who had the illness, the different medications, et cetera, upon the synapses?
Yes, well, that's an excellent question. If people were to look in the article by Bundo et al. That was published just last year while we were in the midst of developing our results, they attempted to address this question in a preliminary way by taking cultured nerve cells and incubating them with low and high doses of risperidone, an atypical antipsychotic, and they were able to show that 2 weeks of exposure to risperidone at either low or high doses had no influence on L1 retrotransposition. Now, it is true that the cultured nerve cell is a long way away from the intact human brain, but this is at least just suggestive of the fact that the antipsychotic drug treatment isn't responsible for this L1 retrotransposition.
Okay, that's an important point, and I guess similarly, the antipsychotic isn't responsible for or isn't doing what we might have hoped it would do, which is decrease the transposition from happening.
Yeah, that's true. I haven't thought of that up until now, but that's a good point.
Right. Well, it's treating the actual symptoms that a person has. It may not actually be treating, and this is no surprise to people living with schizophrenia, for example, it's not actually treating the underlying disorder itself. Some intervention that could directly affect that might actually be something that is treating the underlying illness, not just the symptoms that result.
Right.
For people who are listening right now, and will listen subsequently because a lot of people listen after in the recorded version, what could you tell them if they have a loved one or they themselves are experiencing cocaine addiction, schizophrenia, bipolar, depression, any of these conditions. What guidance would you give somebody right now if they're experiencing these conditions?
Oh, well, if at all possible, the person experiencing symptoms should be seen by a psychiatrist who's best able to assess the nature of the symptoms and provide advice about treatment. That's the single most important thing. And oftentimes, unfortunately, the ill person doesn't recognize illness or isn't interested in treatment. That's certainly true for a lot of people with cocaine addiction and some people with schizophrenia. And so it falls to the friends and family of these people to do what they can to convince the affected person to start treatment.
I think that's a very important point. People should not suffer in silence and should be encouraged to seek help. One question is, with the increasing legalization of marijuana in various places around the country, has there been any studies looking at the effect of marijuana on the brain in the way that you looked at the effect of cocaine?
To our knowledge, no. That would be one of the types of experiments that should be done. I might add that the Okadara group, which incubated these cultured nerve cells with either cocaine or methamphetamine, and found that these stimulant drugs increased L1 retrotransposition in the nerve cells. They also tried ethanol and found no such increase. Unfortunately, they didn't study cannabis or, let's say, heroin or nicotine, and so those studies remain to be done. I suppose it is essential for us, if the cocaine work can be confirmed, to extend this work to the examination of the post-mortem brains of people who died of these other kinds of addictions.
I agree, and I think that this is just wonderful that you're able to share with us such cutting-edge research that I think has the potential to have a tremendous impact. Wade, I want to thank you for a very informative presentation and for the work that you've been doing on this important area. I also want to thank go ahead.
Well, thank you for giving me the opportunity.
Good, thank you, thank you. I also want to thank everybody who's joined us today. All of the research that we fund is made possible through private donations. So if you'd like to make a gift, please visit bbrfoundation.org or call 800-829-8289. This webinar has been recorded, so if you've missed any portion of the presentation or would like to share it with a family member or friend, visit the webinar page on our website. I hope people will join us again next month when Dr. Matthew State, Distinguished Professor and Chair of the Department of Psychiatry and Director of the Langley Porter Psychiatric Institute at the University of California in San Francisco, will present a webinar entitled Autism and Genes. This will take place on Tuesday, March 3rd at 2 o'clock Eastern Time, and I'm just reminding people, usually we do this on the second Tuesday of the month, but in March we'll be doing it on the first Tuesday, March 3rd. Once again, thank you everybody for joining us, and I want to wish everybody a pleasant rest of the day. Take care.
Thanks, Jeffrey. Bye-bye.
Thanks, Ray. Bye-bye.